Welcome to another Ask X-Site. I'm Tim Bushnell with Expert Cytometry. Today's question comes from Jacob. And Jacob asks, what is imaging flow cytometry? Now that's a great question. Imaging flow cytometry is a hybrid technology that combines the power of microscopy with the speed and measurements of flow cytometry. Now, the first imaging flow cytometer was published in 1979 by Wheeless' group out of the University of Rochester. But it took a little bit over 20 years for the technology to get caught up to a place where it could be commercialized. And the first instrument, called the ImageStream, was released in the early 2000s, and it was able to measure 6 multispectral images using a CCD camera and a proprietary time-delayed integration, along with a method to focus the cells so that the system knew where the cells were and was able to measure cells in flow. And with current technology, the latest generation instrument is now able to measure 12 multispectral images at 500 cells per second or more. Imagine now the power of being able to see the cell and see the fluorescence and be able to do a whole host of new assays, from phagocytosis assays to nuclear translocation assays to co-localization assays. The ImageStream and the imaging flow cytometry technology opens up a host of new ways to look at cells and cell processes. And this technology is continuing to be expanded with new markets and new techniques and new instruments coming into the field. So if you want to combine morphology and flow cytometry, think about imaging flow cytometry. Thanks for another Ask X-Site. Until next time, be sure to keep up with your flow cytometry best practices right here with your friends in Flow, Tim and Team X-Site.